This is Emily the fox. She has a beautiful red coat because of the DNA in her cells. However, Emily doesn't quite understand how DNA can control traits, and she is very curious, so I'm going to help her. DNA is like the instructions for creating living things, such as the making of proteins. Certain lengths of DNA are called genes. Each gene contains the information needed to make one protein. However, DNA cannot make protein without help. Consider DNA to be like a master plan for a building. The master plan is not taken to the work site. Instead, a blueprint copy of the needed page is taken to create a part of the building. The blueprint can be compared to RNA. RNA is a copy of one of the template strands of DNA. The DNA is transcribed onto RNA through transcription. Then the RNA is used to code for proteins. This is the central dogma of biology. DNA to RNA, RNA to protein. The first step in protein synthesis is transcription. This is when RNA is created from a DNA template. There are 3 steps to transcription. The first is initiation. In initiation, an RNA polymerase attaches to an area on the DNA called a promoter. The promoter tells the RNA polymerase where the beginning of a gene is. RNA polymerase is an enzyme that creates the RNA. The next step in transcription is elongation. In this step, the RNA polymerase creates a complementary RNA strand from a strand of DNA. Only one strand of DNA is copied. This strand is called the antisense or template. The strand not copied is called a sense strand. RNA polymerase acts as a helicase as it unwinds the DNA, but it also creates the RNA. In elongation, the RNA strand is completed. RNA is different from DNA in 3 main ways. First, instead of having the base thiamine, RNA has uracil. Second, RNA has only one strand. And third, instead of deoxyribose, it has ribose. Because of this, RNA has uracil, adenine, guanine, and cytosine. 3 of these bases in a row is called a codon. Codons will be very useful in translation, but before that, the last stage of transcription is termination. In this step, the RNA polymerase hits the terminator, which signals the end of the gene. Then transcription is ended and the RNA polymerase lets go. But before the mRNA can leave the nucleus, it has to be processed. mRNA is composed of introns and exons. The introns don't code for anything, so they must be taken out. The first thing that has to happen in editing is the removal of these introns. Then the remaining exons are spliced together. And last, to finish the mRNA, a backwards guanine cap is added to the 5' end, and a polyadenine tail is put onto the 3' end. Now that mRNA is mature and ready to go out of the nucleus, in the cytoplasm of the cell is where translation occurs. Translation is when a protein is synthesized using the information in the mRNA. It also has 3 steps, like transcription. The first involves the ribosome and mRNA. rRNA helps to compose the ribosome. It is made of a small subunit, a large subunit, a P site, and an A site. In the mRNA, the first codon is AUG. It is called the start codon because it tells the ribosome where to begin creating the polypeptide. The ribosome binds to the start codon. The first tRNA comes into the P site. tRNA carries the amino acid that corresponds to its anticodon. The anticodons in the tRNA correspond to certain codons in the mRNA. In this way, the correct amino acid sequence can be created. After the first tRNA has occupied the P site, then the next one comes into the A site. Afterwards, the ribosome joins the amino acids together in the P and A sites. Once the amino acids are bonded, then the tRNA in the P site exits and the ribosome moves down a codon, allowing an empty A site for the next tRNA to enter. This assembly line to create the polypeptide is the elongation, the second step. Last is termination. Once the ribosome comes to the end of the mRNA, it hits one of the stop codons. The protein is completed and everything is released. Now back to Emily R. Fox. She understands how the protein is made, but how does that make her fur red? Proteins control traits in organisms, such as Emily. However, she is curious about her friend Lauren. Lauren's species of fox is only supposed to be red, so how is she black? This is probably due to a mutation of her genes. A gene mutation, such as a deletion, changes only one gene. However, if that gene controls an important trait, then it will change that trait and possibly cause problems. Let's say a deletion happened in Lauren's gene that controlled fur pigment. Because one of the bases was deleted, all the codons are different after this mutation. Because of that, the amino acid sequence is altered, which in turn also alters the shape of the protein, which alters the function. This change could make her fur different, such as black instead of red. Emily and Lauren are so glad to finally know why they are the way they are. It's all because of tiny proteins.